a mentor who can introduce you.
Oh, yes.
Yes, of course.
Can we be loud now?
Yes, I think it can be loud again.
This morning we're going to be quiet. My name is Ashley Turner. It's my honor to introduce Taylor Mabry. She's an undergrad in the wormhole research lab that I lead in the Department of Biology. Taylor and I connected over a year ago, and she was very persistent about, hey, I need to be in the lab. Hey, I need to do research. Hey, I need to be in the lab. So curtail time. She's now a senior and picked up a project working on systemic lupus. So Taylor, take it away.
Awesome. So, my presentation is going to be on the genetic assessment of systemic lupus erythematosus associated with the TREX1, or I say T-Rex, variant of uncertain significance and C. elegans. And this is a C. elegans right here. It's as small as an eyelash. So, Jordan Peters also worked on this, Marli, Auslyn, and of course Dr. Turner as well. So, just to begin, due to evolutionary relatedness between humans and nematodes, C. elegans can serve as a model to assess the functional impact of variants with uncertain significance, which is BUS. Just to let you know. And then TREX1 is a gene that encodes to the 3 to 5' DNA exonuclease, and its loss of function of the TREX1 causes the accumulation of unneeded DNA in the cytosol, and these fragments are recognized as foreign material and activate an innate immune signaling. So this here shows the information in the INF alpha and all that situations there. And then TREX1 variants have been associated with systemic lupus erythematosus, SLE, and cardiovascular syndrome. I hope I said that right, I've been practicing that all day. And in this study, the conservation of the TREX1 bus gathered them to determine potential and functional impact. And just some more on SLE. It's the most common form of lupus, and it's a chronic condition that occurs when the body's immune system attacks your own tissues and organs. So you don't want that to happen, just to put that out there. It causes widespread inflammation in the tissues and often affects the joints, skin, brain, and lungs. That's digestive tract, kidneys, and blood vessels. And then AGS is a very rare inherited disease that affects newborn infants, and these two forms show an early onset that is severe and or late onset that has less impact in neurological function. And so then our hypothesis is if a conserved bus is identified in TREX1 and its nematode ortholog WOTF12.4, then it will impact the function and lead to extended lifespan in the nematode compared to the control nematode. And then we use ClinVar here in order to find a variant. So this is our variant that we used here. And then we use benchling to see the MS and your multiple sequence alignments for TX1. So you see here it's conserved all around the board with the human nematode, mouse, zebrafish, cow, and pig. So there's that. And then we're going on to the polyphen 2 predictions. And this is a bioinformatic tool that shows the impact of nonsense mutations in the protein and their pathological effects. So here we see that the TREX1 is in the red, so it's very pathogenic here. And then we have our primers. So the goal of a primer is for the DNA to amplify at various temperatures in the machine in order to narrow down the proper temperature for PCR to run at optimal thing in order to achieve the expected band product size. So you see here that the temperature gauge here, I don't know if y'all can actually see it, but it's 65 to 55 here. And you see that the bottom band here is still showing. So in order for that to disappear, we increase the temperature gauge here. And so it goes to 65 to 75. And so that just shows that the primer set 2 produced better results than primer set 1. And so then primer set 2 will be used in future experiments. And the PCR product including primer set 2 ran at 67.4 degrees here for F2. And it's the optimized PCR that we'll continue to do experiments with. And then the next step will be PCR product for screening and Michael Jackson's CRISPR experiments. And this is where I just keep going. And CRISPR is basically just a new gene editing tool or technology that allows researchers to go into any species genome and make changes. And this will explore the potential impact of bus when placed in the C. elegans gene. And then the discussion here is the T-Rex1 bus is highly conserved across the species. There. And the polykinetic presented to the bus to be probably damaging the score of 0.982 and using predictive modeling. And primers have been optimized to amplify bus in the nematode, and activity of the Cas9 enzyme will be tested using PCR-based assay and an in vitro CRISPR Cas9 activity assay, and that just means like in a medical setting, not like in the body. That's vivo. And then CRISPR Cas9 reagents will then use injected into the nematode to generate the bus C. elegans model. So basically, what this is talking about here is that I'm going to go and redo this here, and then use this, use the CRISPR and everything to find basically the break in the nematode mutation here, just so that we can find out what the restriction enzyme and cut that. I'm cutting that site there. And the conclusions here: the conserved T-Rex1 patient bus is supported in further in vivo assessment, and further experiments include the in vitro CPR, CRISPR Cas9 activity assay, microinjection of CRISPR Cas9 reagents, and the nematode for gene editing, and W02F12.4, and mutant screening to identify the bus C. elegans strain. And thank you to Dr. Turner and everybody here for listening to me, and I hope y'all have a great day. I hope I make sense this way.
Okay,
Yes.
okay.
Yes, sir.
So I understand that we're trying to figure out if something, a certain gene or a certain, whatever the words are,
Get you.
can be bad for us, right?
Right.
And so we can do that by testing it on this little elegant person up here we have.
Yes.
But I don't understand what we are testing to see if it's bad for us. What's systemic lupus erythematosus?
So basically, it's just like, what is the disease, basically, or?
So, okay, you're saying the variants that are caused by the disease, so if I have it, I'll get those?
Yes, so it's a possibility that you can. And
So if I'm born with it, or if I get it after I was an adult or something, I can still have it?
you're born with it, yeah. So
Okay.
it's like, yeah. So, it's
So
like
only if it's congenital, is that what you're saying? Okay.
Yes.
Okay.
Any other questions, maybe? Yes.
So whenever you do the CRISPR, are you expecting anything to happen to the nematode, like shorter lifespan, anything like that, or?
Yes. So, like I was trying to say here, I guess I didn't explain that. It should have a lower life expectancy, just because if you have something damaging your tissues and going after all the basic life functions, that you would see a decrease in your lifespan there. And yeah, so it should be. Yes.
So why are you using nematodes instead of a different model?
Just to be frank, in the layman terms, they're cheap and small. So that's fine. You're just able, just in case, because they're so tiny, and when we do microinjections, it's very easy to, stick something where you're not supposed to be sticking it. So it's just be able to have a little, another one on file. Just be able to do it that way.
So when you say you do microinjections, are you doing this with like yourself manually?
Yes.
So you're under a microscope poking a tiny, tiny needle into something that's the size of an eyelash.
Yeah, it's like I don't know if it's like a little cast like needle sort of situation there, and it's like really, really tiny. And then you go underneath the microscope and you go.
Are you driving it with a robot, or are you doing it by hand?
By hand. Yeah. Yes, by hand.
How?
Practice.
Make sure you don't drink caffeine.
Yeah.
Don't
I
have
say,
caffeine that morning.
yeah,
Yeah.
steady hands.
Jeez.
So like that's why cheap and small.
Yeah.
So like just in case, like just putting that out there. Because
Okay.
Any other questions?
All right. Well, let's thank our speaker.